Hi students, today we have to study about sexual reproduction in plants. Sexual reproduction involves the fusion of gametes, fusion of male and female gametes. What are the reproductive structures of a plant? Flowers. Flowers, the structure we have studied in 11th standard. Still we will be revising that just to recollect your memories. We will revise. Flowers are the special reproductive structures of a plant, and in sexual reproduction fusion of male and female gametes take place. What are the processes involved with the sexual reproduction? Meiosis take place. This also we have studied in 11th standard. Meiosis means reduction division, where a diploid cell divides into haploid cells. Here, for the formation of gametes, from a diploid cell, haploid gametes will be formed. So this meiosis is involved in sexual reproduction, one thing. Then what is the another thing? Fusion of male and female gametes. 1 minute. Yes, fusion of male and female gametes, that is known as fertilization. Okay, this fertilization leads to the formation of a diploid zygote. So from haploid gametes, a diploid zygote will be formed. Both haploid and diploid, the terms it is familiar to you. Haploid means single set of chromosomes. Gametes will be having single set of chromosomes. So male gamete will be haploid, female gamete will be haploid. Both together fuses and form a diploid, two sets of chromosomes. It will diploid zygote. Okay. Then embryogenesis is involved in sexual reproduction. The zygote will be developing into the embryo. That also we have to study. Then what it leads to? Sexual reproduction leads to the production of genetically dissimilar offsprings. Naturally it will be dissimilar than the parent cell, because two gametes, male and female gametes, are involved, and that way the genetic makeup will be not exact same like parents. There will be some differences. So genetically dissimilar means offsprings are formed. Okay. Now all together we can divide the changes or the processes involved in sexual reproduction into three distinct stages. Pre-fertilization, that means before fertilization what happens. Then fertilization, and the third step, post-fertilization. After fertilization what happens? So in all these three steps we will be discussing. Okay. Now we will go through the structure of flower in detail way we have studied in 11th standard, but still this is the stalk of the flower, that is known as pedicel. Okay. And after this pedicel there is a base on which all the floral whorls are arranged. This base or floor, it is known as thalamus, on which the four floral whorls or the floral parts which are having four layers or whorls that are arranged on this thalamus, it is known as receptacle also. Okay, thalamus will be actually familiar to you more because that term is given in your book, but it is also termed as receptacle. Okay. Now on this thalamus all the floral whorls means it is they are arranged. What is the first word? It is calyx, this green color calyx. You know calyx, it is green color and it is made up of sepals. Calyx is made up of sepals. Okay, each part of means calyx, that is a sepal. Calyx made up of sepals, then the colorful portion, what it is known as corolla. The second layer, corolla, it is made up of petals. Okay. Then the third one, that is andrecium. Andrecium, it is the male reproductive whorl of the flower. It means each part of an andrecium, we can call it as stamen. One stamen consists of anther, filament, anther, here this yellow color anther, and this means long stalk, that is filament. Filament and anther, and anther lobes will be connected by connective. Okay, that term also you know. So anther and filament for stamen. Now the innermost whorl of the flower, that is known as gynoecium. Gynoecium, it is made up of carpel or pistil. Okay, carpel or pistil. What are the three parts of means this carpel? This swollen portion, this is the ovary. Ovary is having ovule inside. Okay, you can see this. Then the long, this thin means long tube like structure, this is style. So ovary, style, and then at the tip there is a stigma. Okay, so three things are there in the carpel or pistil. Ovary, which encloses ovule. Okay, here. Then style and stigma. So these are the means floral parts. So in sexual reproduction, we have to study in detail about andrecium, which is means the male reproductive whorl. Then and gynoecium, the female reproductive whorl. In that more detail, we have what we have to study: how the male gamete it is produced in the andrecium, or exactly in anther, and how the female gamete is produced inside the means ovule, in the how the female gamete will be produced. These things we have to study. An activity given in your book. This is the activity. Actually, the structure of the flower is given, and the parts, it is not labeled. You have to label. So already you know the structure. So this you have to do it, and in examination also this way. Unlabeled diagram will be the diagram which is not labeled, it will be given, and you have to label the parts. That way the question can be asked. So this is an activity which you have to do. Okay, we have to study the first the structure of anther. It's how the pollen grains are developing. Pollen grains or male gametes or microspores are developing. So before going into the structure, the formation of microspores, we have to see the structure of anther. Generally, anther is dithecous. Dithecous means two lobes are there. Generally, actually, most of the anthers, they are dithecous with two lobes. Each lobe will be having two pollen sacs. So total there will be 4 pollen sacs or microsporangium. There will be 4. So that way they are tetrasporangiate. Two lobes, it is known as dithecous. Then there will be total 4 pollen sacs or microsporangium. Okay, then it is 4 means tetra means 4. So tetrasporangiate. Okay, 4 pollen sacs will be there. Some monothecous anthers are also there. There only actually 2 pollen sacs will be there. Only 1 lobe and 2 pollen sacs. But generally anther is dithecous with 2 lobes, and that way 4 pollen sacs or 4 microsporangium. Now immature anther, when we see immature anther means it will be having a group of parenchymatous tissue, and it will be surrounded by a single layer of epidermis. Okay, that way the immature anther, its structure. When it start maturing, that is the differentiation, it is starting, that time some hypodermal cells just below the epidermis, some hypodermal cells, they are transformed into archisporial cells. This term will be new to you. archisporial cells. So these cells, each archisporial cell will be dividing into two. Okay, there will be an inner sporogenous cell and outer primary parietal cell. So once again, during differentiation, the hypodermal cell, some hypodermal cell, what they will be transformed into special archisporial cells. It is the starting of the maturation of an anther. Okay, and each archisporial cell will be dividing into two cells, that is inner sporogenous cell and outer primary parietal cell. Okay, now sporogenous cell, what happens to this? The sporogenous cell will be dividing, and the sporogenous cell will be dividing to form a tissue, sporogenous tissue. Okay, and in this tissue again there will be number of cells. Each cell is capable of giving rise to a microspore tetrad. Tetrad means actually 4. You know, the during meiosis, one cell divides into 4. So during gamete formation, from a single cell there will be 4 gametes developed during the formation of male gametes in plants. So that process we will be discussing in the next video, that is microsporogenesis. So just remember in your mind, it is a tetrad. A sporogenous cell, it will be multiplying to form a sporogenous tissue. Each cell in this sporogenous tissue, it will be capable of giving rise to a microspore tetrad. Tetrad means which contains 4. Okay, 4 means gametes or cells, whatever, it will be 4. So from each cell there will be 4 cells forming. That is why it is known as a tetrad. Now what happens to parietal cell? The parietal cell, which is the outer one. Okay, parietal cell will be dividing to form anther wall layers. There will be 4 layers in the wall. So we have to see what are they. We will see one by one. There are 4 layers for the anther wall. That is, outermost one is epidermis, then second one is endothecium, third one is, they are the middle layer. Middle layers, we also, it will be level because there can be more than one layer also. So middle layers, more correctly, 1 or 2 layer will be there anyway in that. And the innermost one, it is tapetum. Once again we will go through that. 1 minute. Okay, the outermost layer, here you see in the diagram, epidermis. Epidermis is actually made up of flattened cells, tabular cells or flattened cells. It is a common wall layer for the entire anther. You can see over here, it is there throughout. It is a common wall layer for the entire anther. Okay, this outermost one, epidermis, it is protective in function. And what is the second layer? Endothecium. See, here it is labeled. Endothecium means this second layer. It is made up of radially elongated cells, and they are having fibrous thickenings. This fibrous thickenings, it is due to the presence of callose, which is a polysaccharide, and these fibrous thickenings actually help during the dehiscence of the anther wall. After maturation, the microspores of pollen grains, it will be released out. During that time, this fibrous thickenings, it is helping. And see, this endothecium, towards this groove, it will be thin layer, and in that area actually the dehiscence or splitting up or breaking take place. Okay, over here, that is the second layer, endothecium. Then inner to endothecium, what we studied. What is the third one? Middle layers. I told you middle layer or middle layers. It can be a single layer, or there can be 2 layers also. Okay, so we call it as middle layers. So third one is middle layers. It is actually specific to each sporogenous tissue. Okay, here it will be there, means middle layers, then here it will be there, here it will be there, and here also it is means peculiar or particular to each pollen sac or sporogenous tissue. Okay, outer epidermis as well as endothecium, it is common for the entire anther, means entire anther. Okay, common wall layer, but the middle layer it will be surrounding means its own pollen sac inside. Okay, here. And what is the innermost one? Means it is the tapetum. Tapetum, it is here, it is the innermost wall layer, which is the means next to the pollen sac. Here, pollen sac is here. Okay, or sporogenous tissue. So just close to that, that will be the tapetum, the innermost layer. It is nutritive in function. It is innermost nutritive layer of the anther wall. It immediately encloses the sporogenous tissue. So here you can see tapetum, here you can see. Okay, so again it will be particular for each pollen sac. Okay, understood the different parts? Outermost one, it is epidermis. Okay, it is a single layer of flattened or tabular cells. Then it is common for the entire anther wall. Second layer is endothecium. It is made up of radially elongated cells with fibrous thickenings. Okay, endothecium. Endothecium will be actually thin in this groove. Okay, thin over here. Here it will be thin, and that area actually the dehiscence take place. You will come to know what is dehiscence in the very next diagram. Okay, and then the third one, it is middle layers. Middle layer is particular to each pollen sac. It will be surrounding. And the innermost layer of the anther wall, that is tapetum, again it is means particular to each pollen sac, and it is the innermost one, nutritive layer, and it is the closest one to the pollen sac. Okay, it is nutritive in function, and it is immediately it encloses the pollen sac or sporogenous tissue. Sporogenous tissue it is given. Afterwards here inside pollen grains will be developing. So we call it as pollen sac also. Okay. Now we will see one more diagram. These diagrams are given, the same diagrams what is given in the textbook, that only is this one, because for examination the diagrams you have to follow what is given in the textbook. Here you can see a dehisced anther. Dehisced anther means what? After maturation of the pollen grains inside the pollen sac, this area it is broken. You can see over here, and see here two pollen sacs were there this side, isn't it? Both the pollen sacs are joined together, and you can see enormous pollen grains inside. The same way this side also, two pollen sacs they were there. They are actually joined to form a single sac, and pollen grains will be released out through this area of dehiscence. Through this area the pollen grains will come out. So here during dehiscence the middle layers actually they get disintegrated. Okay, and the endothecium will get thin in this area. It already said that groove area it will be thin, and through that the dehiscence take place and the pollen grains are released out. So in diagram, if it is TS of anther asked, then the before diagram you have to draw. If the dehisced anther is asked, you have to draw this diagram. The diagram, if they will be particularly mentioning, then dehisced anther you have to draw this diagram only. Okay, so if it is asked TS of anther generally, then the first diagram you have to draw, and TS of anther dehisced anther if it is asking, then you have to draw this diagram. They are actually each side there will be a single sac, you can see, because the middle everything it is getting disintegrated and pollen grains can come out through this area. Okay, so that way you have to draw. That is the structure of anther. And now we have to study the microsporogenesis or formation of pollen grains, what it is happening, the structure of pollen grain and formation of means microspores or pollen grains. So that I will be means explaining in the next video. Okay, so this structure you study the very next video, the formation of pollen grains or microspores, that is microsporogenesis, I will be explaining. Thank you children. Okay, bye. Go through the video and the parts also, means it will be, the diagrams are new to you, so keenly observe the diagram and study and give me the feedback. Okay, bye. We will see in the next video.